My channel, my channel, my content. My channel, my content. My channel, my content.
Happy Monday.
Hello, everybody.
Hello, happy Monday.
There we go. And Heather?
Hi, Billy.
Happy Monday.
There we go. Everyone's here. Yes. So thank you guys for coming. This is, pretty exciting for me, and I know for many people. I know it's been talked about, this subject, and I think getting everybody sort of involved together at once and sort of talk about what we're doing and, where it can go, what, the positives and negatives and all that. I think it's pretty awesome. So I guess what we'll do is we'll start. Everyone knows me. Everyone knows Austin. Go ahead, introduce yourselves, and then Heather, you can take the lead and let me keep my yap shut for a while.
Sure. So I'm Heather Roffey. I'm based here in Montreal. Go Habs. And I am a small, modest ball python breeder. You can see pretty much my females right there, back there. And I'm actually also, as my day job, I'm an biology instructor at a local college here in Montreal. So I talk about genetics all the time in my classes. So to be able to merge the two is just so exciting for me.
Go ahead.
Hi, my name is Hannah Seidel. I'm a professor at Eastern Michigan University, which is near Detroit in Michigan, and I'm a geneticist by training. I don't own any ball pythons, and I've been involved in understanding the genetics of color morphs and using that in a classroom setting to help teach undergrads techniques in genetics.
Awesome.
And maybe I'll go next. I'm a PhD student. I'm Alan Garcia-Elfring. I'm a PhD student in the Barrett Lab at McGill University. I have had a ball python. It was just a normal, but I thought it was still pretty cool. I've also had a milk snake, as well as tons of garter snakes and frogs when I was a little kid.
Awesome. Last but not least.
And I am Chiron Graves, and I work with Hannah at Eastern Michigan University, primarily working with future science teachers. So I teach teachers how to teach.
Awesome.
So that's one of the things that I'm trying to incorporate into this, the mix, the work that Hannah and I are doing at Eastern Michigan. Heather mentioned that she's a small breeder. I'm way smaller than she is. I only got 20 back here.
Yeah, you know what? You can never start too late, and just having them is just awesome. We got to get Hannah one just to
Oh, I'm working on that, Billy.
Good man. So like I said, welcome all. And Heather, you want to just sort of take away sort of like a relapse of how we're all connected in some sort of way, shape, or other?
Yeah, so this project actually started a number of years ago. And it actually started at a holiday Christmas party that we had for McGill graduate students and some professors. And it was kind of a show and tell. I brought out all my snakes, as I'm sure many of you guys like to do when you have friends over. But one of the people that was there was a professor, Rowan Barrett, and he has studied the genetics of coat color in mice. And we talked about how there's nothing been done with ball pythons. And so, of course, the little scientist's brain started going. And we had discussions about this, and we're like, you know what? We can do something with this. We read that you can extract DNA from sheds, so we reached out to breeders that could get us here in Canada, get us sheds. That's where Billy comes in. And then we're like, you know what? We really need a graduate student on this because they get things done. So enter Alan. And then interestingly, running just at the same time, so this is actually around the summer of 2017, we get an email from Hannah, and she had seen our interest with posting for a graduate student, and she had the same idea involving, she and Chiron involving undergrads and doing ball python genetics. So yeah, and it's awesome that we're all here with stuff to report 5 years later.
Yeah, no, it's been a crazy little journey, and I just think it's so amazing. And when I thought of it, I was just like, wow, the possibilities here, especially me being the freeway guy and having to deal with asphalt/yellow belly for 12 years now, and it's like I can't tell. I thought I could. But then every one that I would hold back, we'd breed. And what do we get, Austin? Oh, it looks like ivory. So that's a yellow belly. So much for my IDing. And it's frustrating when you're trying to create something and you don't know if you have the right genes. So as a breeder, I've always had to hold back and keep stuff back and trying to, hit the long shots with the genetics in those animals. And if they're not in there, then my long shots are really long. And then from the industry side, I've been burned. Many people are getting burned. You're buying an animal because of the genetics. And, I don't think many people do it on purpose. It's getting really hard to ID animals because of the amount of genetics and polymorphism. You can see one animal and they have the exact identical genes and they look totally different. Oh man, this one's brighter. Maybe it's fire. Maybe it's vanilla. It's so strange how they come out and you're perplexed. And, I'd like to get it to a genetic, DNA testing where it's like, hey, this is what it is. That's just a nicer version. But I hate seeing people get ripped off. I would like to know what I'm selling. So if I'm selling something, there's a few people that do super fighters. They're a white snake, and it's possible Enchi, possible OD, possible this, possible leopard. We're selling an animal that has the potential of being a $2,000 snake for $500 because we can't tell. And then again, if we knew it had all those genes, we probably wouldn't be selling it at all. So I think it's great for the future of this industry to sort of know what we're selling, what we have, and I think it'll be a little bit easier, especially for breeders, when we're doing triple, quadruple, recessives. When we miss, and don't hit a visual, everything's possible 66%, and then we're trying to breed possible 66% to possible, it's just nuts, So I definitely love where this is going and what's going on, and Hannah, I actually heard one of your podcasts that you did on Friday, and I learned a lot, what you guys were doing and how you're doing it. Can we explain how we're doing it and how it's, unique or how it's different, Alan or Heather, how, ours is a little different and, how they are both, complementary to each other, or maybe they're not. I'm not really sure, but I'm pretty sure they are, right?
Yeah, I can...
Yeah, well, I was going to let Hannah explain what she's doing because I was doing all the talking, so go ahead.
Sure. So we've been doing what we call a candidate gene approach, which essentially means that we make an educated guess about what gene we think might be causing a certain color morph. And that educated guess comes from information that we learn in the scientific literature from what's known about genes that control color patterns in other animals. So typically that information would be coming from studies in mammals or studies in fish are the most common. And then we sequence those genes in animals that are a particular color morph and in animals that are not that color morph, could be normals, could be other animals that are just simply not the morph of interest. And then we compare those sequences and we ask, are they different? And are they different in a way that would make sense in terms of disrupting the function of the gene?
Awesome. Yeah, I get most of it, but it's still so hard to comprehend too. It's like, oh, cool. You make it sound so simple and easy, but it's not. And it's just like, oh, you do this. It's like most of us are sitting there going, okay.
Right. Okay. So I guess I glossed over the part where we collect sheds from people. So the first step is to go on Facebook, go on Twitter, go on Instagram, tell people we know, go to local reptile shows and just publicize that we need samples, and we need samples from everything. And then we collect samples and then we extract DNA from the samples. And once we've got the DNA, that's at the point where we do the DNA sequencing. That's when we sequence particular genes in particular animals that we think might be causing a particular color morph.
Okay. Now, how is, the way you're doing it, Alan, different?
So the way we're doing it is, like Hannah, we're also extracting DNA from shed skin, but instead of targeting a particular gene that we have, at least, an educated guess that it might be involved in that particular phenotype, we kind of go in just ignorant of wherever the mutation might be and sequence the whole genome, so it independent of a gene. And so the trade-off is that you don't need to have any a priori knowledge of what potential gene might cause a specific phenotype, but you're sequencing the whole genome, so it's quite a bit more costly. And so, yeah.
And you did this with, one of my favorites.
Oh yeah, sorry.
Piebald.
Yeah, with piebald. So maybe I'll give a little bit of background. So I did my undergrad in Vancouver Island, and then I came to Montreal to do my master's. I did it in a different lab. And then I wanted to join the Barrett lab because they were doing cool stuff like pigmentation. And then I saw this tweet from Andrew that there was this opportunity to study ball pythons. And I had actually got into biology because I was one of those kids that really loved reptiles and stuff. I used to collect tarantulas as a kid. I was born in Mexico, so I used to just go out my front yard and start digging up tarantulas. But in undergrad, you hear that all these cool animals, or at least the ones that I thought were very cool, you don't actually do research on them, at least not like genetics. You hear about model organisms, right? About doing stuff on fruit flies or on nematode worms, as Hannah did. So I was very excited when I saw this opportunity to actually merge my two passions, right? Genetics, which I acquired during my university training, and reptiles and snakes in particular. And so, yeah, I took this opportunity up. And so the first morph that we wanted to target was a recessive, was I think one of the first ones in the pet trade, piebald. And so, yeah, the approach we took was pretty much sequence around 50 individuals that are piebald visual, and compare them with 50 individuals that don't have piebald, that aren't even het for piebald. And then this will give you the genomic region where the mutation should be. And then we look closer in this particular genomic region to look for candidate mutations that might explain the phenotype. And as you guys might have seen, so we published a preprint and we found a premature stop codon. And all that means is pretty much that it stops the protein before it's fully synthesized. And so you have a non-functional protein.
So now, how long did it take you to discover this, the whole process, and how much time? I know it wasn't easy. It wasn't something like, oh man, this is so, I should have done this years ago. It was definitely a challenge, and you definitely rose to the challenge and nailed it.
Yes, well, one of the issues is that the ball python doesn't have its own reference genome, and so we have to use the reference genome of a similar species, a related species, the Burmese python, which I think all python enthusiasts will recognize. So that's fully sequenced, and it's annotated. And what annotated means is that the genes are mapped to a particular sequence. So for gene A, it'll tell you that on this part of the genome from position X to position Y is this gene. And so I had to find which genes might be a candidate mutation, and then also finding which gene might be the candidate gene, and then from there finding the candidate mutation. And I had to look for there was actually one thing that slowed me down is that the reference genome is highly fragmented. And so I wasn't even sure if my data made sense because I was getting peaks all over the genome. And it wasn't until I aligned it to a reference genome that came out recently, that's actually chromosome length, that I saw the actual region in the genome, that it's only a single region of interest. So that kind of pointed me to the right direction.
You must have felt so good when you saw that peak.
Yes, I'm like, oh my God, I'm not useless sort of thing. Yeah. And then I discovered this software that allows you to annotate SNPs. And so another thing that slowed me down is that, again, because it's not a model organism, this software didn't have the reference genome in its sort of database. So I had to manually add the ball python reference genome to it and then run the program. But once I ran the program, I had a candidate mutation pretty quickly. And so I think now with subsequent morphs, we could run this protocol in pretty quickly and get the genetic basis for a bunch of morphs.
Awesome. So you're saying that, because even at the beginning, it was all like, yeah, it should be possible. It should be possible. Now we've proven it possible. Now, where do you think, how long do you think it will be to crack all the, I guess that's not the right word, but discover all the right genes and where they are and the sequence, the location? Is it something that's going to be easy moving forward? Are they going to be all over the place and each one is in a different region? Is it going to be something where you can sort of duplicate your process that you've already done? Or is it like starting all over from scratch, but you have a better idea of where to look?
I think we can replicate our process pretty quickly. There might be some mutations that might be harder to pick up, like for example, a large indel, a large deletion of the genome. With my method, that might be difficult to pick up, but there are ways to also try to check for that. But I think many of the mutations should be of the type that I and Hannah have found. So mutations that change the amino acid sequence, mutations that sometimes just cut the protein short. Perhaps it mutates the start codon such that the protein doesn't even get synthesized. These sort of large effect mutations that are of just like a single base pair change are likely to be the genetic or underlie. I won't say most because I'm not sure, but I think quite a few of the color morphs.
I just think it's awesome. Like the way you're talking is like, oh, the amino acids and this. And I'm just like, man, it's way above my pay grade, or my scope. But it's just awesome how both of you, I have a passion for snakes and helping people and sharing information. You guys have the same, in the scientific realm and the genetics and it's exciting. And what you guys have done is, I think, is just amazing. I can't understand it all, but it's just amazing. And I think it's, hats off to both of you and everyone else involved that, doing this and for two different reasons, right? I mean, you're doing it for learning and having people, undergrads to do this and learn and study in a cool way. And it's just, I just love it. I just think it's amazing. And, thank you guys for all your hard work and what you're doing. And, hopefully the future is bright and, it just works, without a lot of stress.
So I just want to chime in and say I definitely agree with Alan that probably a lot of the morphs are going to be really easy to identify. And then there's probably going to be some morphs that are going to be much harder. And we don't know ahead of time which ones those are going to be. And typically it depends on what type of mutation it is and basically how easy it is for us as geneticists to recognize that mutation is something that's going to affect the biology of the animal. So sometimes when I talk about this project, I feel I have a concern that people are going to get impatient, when they send me a shed and they say, test this for such and such once you come up with that test. And it's possible that for some morphs, we might have answers within the next 6 months, within the next year. And then for some morphs, it could be years and years and years. So definitely want to say that people, should be patient. It's not for lack of trying. There's factors that we can't always control. Yes,
definitely. And, nothing that is, I think, incredible is ever easy. And, it's something that, just what you guys done already, I think is just mind blowing. I mean, it's just so awesome.
Although I will also say that a lot of things we're doing would have been possible before these last few years. It's just that nobody really happened to do it. So I guess I just want to point out that a lot of this is in fact somewhat simple techniques, and it's really now that ball pythons are becoming more recognized as geneticists as a really useful tool for understanding color patterns in reptiles.
Which is awesome, man. I think, yay, ball pythons.
Yeah, and I know that there's other species of reptiles that also have a lot of color morphs, like certain species of geckos, for example. And some of what we learn in ball pythons might eventually help to identify the genetic basis in other reptiles as well, because probably one reptile to another, or at least lizard and snakes, are more similar than, say, one of those animals to a mouse or one of those animals to a fish. So understanding ball pythons will help us make better predictions for other reptiles too.
Oh, amazing. I didn't even thought of that. That's really cool because, I mean, there's pattern mutations and color mutations in pretty much everything. So
yeah, there's recently a paper that came out looking at a particular morph in leopard geckos called lemon frost. It causes a cancer from the skin. The animals have a particular cell type that humans don't have that forms a tumor. And I've been corresponding with the author on that. He's studying some other leopard gecko morphs, and I basically said, look, I think it's one of these 8 genes. Just look at one of these 8 genes, and I bet you'll find it. So
that's super cool. I love that, you know what I mean? One thing can unlock and help other in areas and other things. I think it's amazing. So cool. So now here's something. I've been, working, for this, like, I think our 4th year, 5th year now, sending in sheds and getting back and forth, a lot of people reached out and, oh, hi, I think, somebody told me that, with a PCI or PCR test, that you can do this from your home, and it's going to be, you have to buy the equipment, and you can do this from your home if you know what you're looking for. Now, do you think that, I've even, anyone can chime in. I sort of talked with Heather like this, about this earlier. Do you think this is something, if we have about 50 morphs that are sequenced and we know what we're looking for, do you think that this can be something that anyone can just do at their house to test? And that being said, can they test, when you test, do you have to test for 1 gene at a time? Or can we send a shed in and test for 9 genes at a time? So, I guess that's a double loaded question. Like, can this be done at home? And do you have to search for, DNA and extract and find 1 gene at a time to test? Or can we do it all at once?
So you, in theory, could test a lot of things at once if we knew a lot of different, the genetic basis of different color morphs. So in humans, for example, there are companies that will test you for, say, 300 different possible mutations that you might carry, that you might pass on to your child, that might cause a genetic disorder in your child. So there's a variety of companies you can pay several $100s to have them perform all these tests at once. In theory, that's possible for something like ball pythons once we identified all of that. That requires a different type of technology than either of us are doing right now. That requires, I think, Alan, you can correct me if I'm wrong, I think they're using some type of Illumina array for that. I don't have to explain what that is, but it's a slightly different technology. But I think that for most ball python breeders, they would typically have just a handful of genes they might be interested in. And so having individual tests for specific morphs would be more cost effective for the breeder, and also might make more sense in terms of the position you might be in terms of, you might want to know, does your animal have any of these 5 things? You might not be concerned about dozens of them.
Okay.
I don't know if I answered your original question. You also said, like, can you do this from home? You probably won't be able to do it from home, but you might be able to send a shed and have it be done inexpensively.
Yeah, because, I mean, I've heard people saying that, and I just want to sort of crush some of the myths, but you never know. In 5 years, you could have something. It's like, oh, here, all you do is, you touch it with this rod, and if it changes color, you got it. Like, you never know.
Well,
I mean, it wouldn't surprise
me.
To be
fair, like, there could be a high school student somewhere who has some of this equipment that he buys or she buys secondhand on eBay, and you could actually do some of these really simple tests in your house if you were going to dedicate a closet and stuff to them. But I don't think a typical person would do that.
Yeah. Well, another thing, I mean, my thing is moving forward, when we actually, we're told, Alan's like, hey, we did piebald, it works, we can actually do this. It's like, wow. I think it was, in my mind, I'm always an investor. I always want to do things for, the best of the industry, and, knowing what I've had to go through for so long with trying to prove stuff out, breeding stuff that I don't know what it is, I'm just praying it is, and to going after these long shots and moving forward, these long shots are, going to be more present in everybody's breeding program. And instead of hitting stuff or trying for a project for 7 to 10 years and figuring out that, hey, man, I don't have the 1 gene that I need, it didn't prove out, it's a lot of time and effort, and it is rough. So to me, I was like, wow, this would be amazing. I think, this would be amazing to have a company in order to process these, send it in, as well as one of the questions, I think it was Alan, is like, do you think there's a need for this? Do you think, that people would be sending in sheds? And I know myself, if the price was cost effective, $20, $30 a shed, and it could be done in a timely manner, not like 5 months down the road, but I mean, if it could be done in a timely manner, once things were moving, I produce, like I said, 18 to 2,000 animals plus a year. And anything that was over $1,000, I would go and pay to have that testing done so that I could sell it as a selling feature, saying, hey guys, here's the animal. It's $800 or $1,000. It comes with a DNA certificate of genetics, so I know what you're selling and you know what you're buying. And myself, just to start, before I even produced an animal, I got about 500-600 animals minimum that I'd want to know that's in my racks, that the genetics that are sitting in there. And I'd also like to find out if you did a test, what would be amazing is you send a DNA or a sample in of the shed, and it's like, yep, it's this, and this, and we have an unknown one. Or, hey, it's also heck clown. What? That'd be amazing, because without being heck clown, you would never probably put it to a clown or a heck clown. Now something pops up, it's like, wow, this is more powerful than I thought. And I think what's really cool is if I was to send something in, and it's like, yeah, it's got 7 genes, but it's got 1 gene we don't have in our database, or we haven't sequenced. We haven't seen anything like it. Where did you get it? Well, I produced it. Cool. Send the shed in from the parents, and then we can find out where that gene came from the mom or the dad, and go from there. I mean, it's endless. But another reason why I think it should be commercialized, or having somebody not doing it in their basement or, in their home, is because we have scammers now, that are taking other people's pictures, or saying, oh, it's het for this or double het, getting money. And then people buy it because we're just, we think everybody in the ball python community is honest and amazing, and the majority are. But there's people out there.
You need like a third person who's doesn't have an interest either way.
Yeah, or to govern it so that it's like, hey, guess what? When we're sending a shed in, it's going to a lab, it's being done properly, and it's being done and sent back with some sort of credibility,
Yeah, so I will say that a lot of the things that make something expensive in terms of this type of testing aren't actually the supplies and the materials, but they're the human who's doing the testing, paying that human, training that human, making sure that human is following the procedures correctly. So the expensive part would be the humans, not probably the supplies.
Well, we can might make a robot for that, right? Come on, everything's being replaced. Humans are being replaced. We'll just make a bot. Just sit there and DNA test, and that's all he does.
So one idea I've kind of brainstormed about, and I'll just throw this out there. So in the genetics laboratory that I teach, this is how I've been doing this project. It's with undergraduate students who are learning about genetics. They're learning about genetic techniques. And I started this project because I wanted to make the class more interesting for them, and also honestly for me as well. And each semester we've done something different because we've moved the project along, and the next step is different than what we did before. And this fall semester, we're going to be doing genetic testing for all of the morphs whose genes have been identified so far. And this fall, I'm offering that for free because I'm trying to incentivize people to send us their sheds. Yep. And the students get the opportunity to learn the techniques to do the test, and then also to communicate their findings to the person who sent in the shed, which is a good experience for them to practice their science communication skills. And some of my students, for example, go on to genetics counseling school, where their whole job is basically going to be doing exactly this, but for people.
Awesome.
And when I've talked about this lab that I do with other instructors who teach genetics labs at other colleges, a lot of them have said, oh, that sounds really fun. I would be interested in doing that with my students. So I have thought about maybe publishing some type of lab that could be widely available, and there could be actually lots of college students all over the country doing these labs where the procedures were very easy for students to follow, and so people could send their sheds to different colleges and have them do different tests. And certainly the student would have no incentive one way or the other to
manipulate,
tell you
or
one
falsify.
thing. Yeah. And here's the thing: you don't have to pay the student, so it wouldn't be expensive in terms of the human part. The downside is, of course, that occasionally students do make mistakes. So.
Now, with mistakes, like, how hard is it to, like, can you, I don't know how it's done, but if you use a test tube and it's not properly cleaned and you put it in, could that, screw up the sample?
The mistakes that students make yes, Heather's nodding. So basically, instructors cannot predict the mistakes that students make. Students make mistakes that instructors, they would never have thought a person would make the decision that a student made. And when instructors do the experiment, when someone who's trained, who has a PhD in some genetics field does the technique, I would say the mistakes are really, really minimal. But students, they don't notice when things don't go right, and so they continue forward, and then can sometimes end up getting the wrong answer, unfortunately.
Yeah, no, sorry, go ahead.
I have tried to assess how often are my students making mistakes. So I've given the same shed to different students, and so far they've always gotten the same answer. I do make a really big deal. So in the chat, Alan mentioned contamination is a potential issue. So what he's referring to there, I think, is if two students, let's say, were working with two different sheds and they were sitting next to each other in the classroom, and they were really sloppy and ended up, throwing a piece of their shed in front of the other person, and that person ended up using that for their DNA because they just weren't paying attention to which pieces came from which animal. That's a really big issue. So when I teach my class, I have students clear off the whole desk, and then I have them take a big piece of tape and put it on the desk in between each student that are, sitting next to each other, and they're not allowed
to cross the line.
Cross the line. Yeah, no crossing the line, because we don't want any contamination. So I make a big deal about that because that is a really big deal.
Yeah, I mean, I know that, nothing is perfect, but, as long as things were followed and it's a procedure, I'm sure things, beforehand you'll see things that's like, oh, we got to prevent this, and you'll see things happening, and then you can correct it to get it to be better. The more common
mistakes that students make don't necessarily result in getting the wrong answer. They just result in getting no answer. So I would be more concerned with just the test failing completely rather than someone getting the wrong answer. And in some ways that's better. It's better to not have an answer than to have a wrong answer, because then you can just do the test again. So that's the typical types of mistakes that students make, result in no answer at all.
Now, I know that you're requesting sheds from multiple people, which is a great way, because, I mean, all we've been doing with them lately is, hanging them to dry or throwing them out. I mean, that's all we do with them. Some people have actually taken it and had they're artistic, and they've made like cell phone covers or rings or jewelry or really cool stuff with some people make soap, with snake shed in it. I mean, it's just so amazing, what, people's thoughts and stuff that we're doing with, even animals, as well as, parts of the animals. So where was I going with this? I get so excited. My brain's just thinking.
Heather's got some jewelry there. She just put
on look at her. There you go.
Yeah.
Hey, I've got a hat from Mary Python, and she made it, and she got some shed, and she basically, what do you call it? Not pigtail it, braided it, and it's pretty cool, man. It looks like it's pretty awesome. So, there's so much things we can do with stuff. It's just, being creative. Where was I gonna go? What I was just thinking of? I do this all the time. I'm hyper. I talk. My mind's racing a million times. Can't get a word in.
We were talking about testing and contamination and samples is what we were talking about.
Oh, yes.
Yeah, I guess, and one thing I wanted to mention too is that you could do the testing this way, where there's a lot of labor cost, where you have to do the PCR. You have to amplify the fragment you're looking for, do Sanger sequencing. So there's a lot of labor to that. But the way that we would like to eventually go, once we have the genetic basis for more morphs, is actually make just like a SNP chip, which is, just like a panel, where each little hole has, I want to use jargon, but
layman's terms.
You can test a particular mutation. Yeah, you can test a particular mutation in every little hole. And so you can pretty much just scan a bunch of individuals with that same chip, and it'll give you, say, three colors for each well or each mutation. Say it'll be green if it's homozygous for the morph in question, heterozygous, say yellow, or say red for homozygous of the normal allele. And so this way you can kind of skip out on all the labor that would work, or that would have to go into testing each individual.
Yeah, if I were starting a company, that's the way I would do it. So if there's any business investors and you're having me as an advisor, that's what I would advise you to do.
So we can have this, what, Alan, by end of the week? Let's do this. Let's get this done.
Or in some sheds.
Yep.
We can push ahead.
So I'll say that technology that Alan described is really similar to, if anyone's done 23andMe genetic testing, that's a very similar type of technology that they use for human ancestry analysis, or testing if you have certain, a variety of genes linked to traits or genetic disorders.
Yep. Now I know what I was going to say. Back to when you were asking a bunch of people to send in random sheds. We make a lot of mistakes IDing stuff where what happens if the animal they send in, I mean, it's not so hard with like albino or like lavender, but some of these other genes down the road, when you're looking for certain things and it's not there, or they're sending in a sample saying it's this, but it turns out it's not. How does that sort of throw off your research, or does it?
So Ellen, do you want to talk about that with your approach, where you probably still have signal because it's a kind of, as long as most of them were right, you're probably still.
I think he's frozen.
Yeah. Oh, so thanks. Although we would want to include. Oh, am I good?
No, you're good.
So if we have a choice, we would like to include individuals that are kind of for sure that particular morph. So in some multi-gene color morphs, the differences kind of might be subtle. So if we were going to include a particular individual in a pool, I think the safest bet would be to kind of be sure that it is that particular morph. So if you have, I don't know, like 4 genes, and one of them, it might be, I don't know, a clown, but you can't really see the clown. I think it would be better to maybe include that individual in a pool of one of the morphs that is totally noticeable, if that makes sense.
Because I know, I mean,
if I remember correctly from your paper, Alan, you had a teeny bit of reads from the wild type allele in your piebald pool, and you guys basically said, okay, that was probably a little bit of contamination, but in theory, it could have been that like one of his 50 animals was actually not piebald, but because the overwhelming majority of the samples were piebald, he was still able to identify the gene because most of the vast majority of the animals were correct.
Yeah, exactly. And so that's also one of the things that took me a little while to find this mutation, because I was expecting it to be fixed, which in genetics terms means that the level of divergence should be 1.0. And so I was looking at all the mutations with this 1.0 reading, and I didn't get any obvious candidates. And it wasn't until I looked a little bit below, around 0.95 to 1.0, that this one popped out. And so, yeah, that's one of the issues. So yeah, we would still get a clear signal, and this is why we can also target spider. Like obviously there isn't any homozygous spiders because it's lethal, but if we have a large enough sample size of spiders, we will get a peak that goes around to 0.05, around halfway up. And then what we could do is just isolate all the SNPs near the top of that peak and look for mutations that affect the protein product. So we don't need we could, it could handle a little bit of noise in our approach.
We have a super chat question. Go ahead, Austin, take it away.
We actually have 2, one pertaining to genetics and then one to morphs, how they look. But let's go with the genetic one first. It's from Octavio Garcia. He says, how will allelic genes work on a test? Let's say we have a chocolate and a spot nose. Will the test show both, or will the results be one or the other in a test result?
So I'll take that because in the analysis of the albino color morph, we found a little bit more complexity than we necessarily had expected. So we analyzed the albino toffee candy complex. So before we started, we thought that there were probably 3 alleles, one for albino, one for toffee, one for candy. And what we actually found is that toffee and candy are the same allele. They've just referred to by 2 different names. And that the albino morph, there's actually 2 different alleles that have been referred to as albino, but breeders haven't recognized them as different. So some albinos have one, some albinos have the other, some albinos have one of each. And because we've identified each one of those alleles, we have a test for each one of those alleles, so we could tell the owner whether the animal has 2 copies of one copy of one copy of another, and so on.
See, that is awesome. That is super cool because, I mean, some of this allelic stuff has just been popping up because we've never really thought of it. You know, we see stuff and we're trying to create stuff. It's like, oh, we got shitty odds. We didn't get it. Maybe next year. And all these people are trying to make something that can never be made. We just figured that the odds gods, that's what we call them, you know, our little thing. It's like, oh, odds gods were mad. You know, it did me great on the other clutch, but it killed me on this one. We try next year. So it's great that we found stuff like this out because people are trying to create something that they can never make because of the allelic animals or the genes.
So you found that that toffee and candy are the same.
We found that toffee and candy are the same, but we also found that not every animal is perfectly identified by their owner. So in other words, sometimes we have an animal where the person said to us, this animal's Toffino. And when we genotyped, it was actually toffee. Or sometimes we have an animal that the person said is Candino, and it's actually just toffee. So there's the designations by owners and breeders don't perfectly match the genetics, although they do more often than not.
You see why we need help? We need help.
So I was going to address the question about, how do we know what we're getting? Early on, I had asked, had thought before, I got somebody like Billy involved where I could get a mass amount of sheds, but I was thinking about asking the owners to be sending in pictures. And I know it's a bit laborious, but I really think that when people send in sheds, they send in a picture. So then if you do see some differences within what we think is the same allele, then you can look at the pictures and start making connections, even though the owners may not know that because they haven't seen other individuals in other people's collections. So pictures would be awesome.
Yeah, more data.
I'll also say that there is some data you can get from the shed. So if you wash the shed really nicely, cut it down the belly, and then lay it out flat on a white piece of paper, you can see the dark areas and the light areas really easily. Like in a normal animal, you clearly see a pattern. So in our case for albinos, we could easily tell that the sheds were essentially an animal that didn't have any black pigment in it. And for something like spider, for example, you'd still be able to see that there was a different pattern. So there is at least some information you can get from the shed.
Yeah, that's like hypo, ghost, orange ghost, hypo. The sheds are clear. When you see a shed, you pour it out. You will not see any pattern. That's a key thing that we do.
That trait is really interesting because it suggests to us what the gene might be, because there are certain genes that are required for transferring the black pigment into the cells that end up in the shed skin. And so the fact that that animal is still making black pigment but is not getting them into those cells, that suggests what genes might be causing that color morph. So that piece of information is really helpful to us as geneticists.
I think it's amazing. Like when someone told me that, I don't know how long it's been since someone told me, I'm like, what? Yeah, you can tell it's hypo. You just do this. And I'm like, no. And you're sitting there pulling sheds and you're like, wow, it's amazing. I still do it to this day. I mean, it's something so minor that we can do that sort of verifies if it is hypo or not. Because when we're making some of these animals, some you can tell hypo right away. And then some is like, oh, I don't know, wait for a few more sheds. And nope, it's not making it any easier. So little things like that I just love because it helps me as a breeder identify and know what I have and know what I'm selling, know what I'm pairing, et cetera, et cetera. So
there was one more super chat.
Okay.
It was from Kane. It says, how do you feel about Orange Dream and Clown? Also high intensity Orange Dream versus the normal Orange Dream.
So have you I don't know if Alan, I mean, I probably Hannah's got this more, or probably ball python people are messaging her with thousands of questions more than yourself because she put herself out there. Have you heard of high intensity orange dream and orange dream?
So Orange Dream is a morph that I haven't focused on so far, in part because I'm less certain about what the morph looks like and what a super would look like. So I feel very unqualified to answer this question.
Okay, yeah, because I know that it's something that people ask me all the time, and I'm still up in the air. I'm like, I don't know. But questions like this down the road, we're going to know genetically how they act, what they are, what they're capable of, how they sit and how they work together. Are they allelic with certain things? And I think this is something that, I think it's great for the industry, but it's going to also hurt some people's egos, as well as we have a thing called trick, hurricane, and blitz. No, they're all the same. And then people that have the certain ones, no, it's totally separate. So when it gets down to, genetics and seeing that they are exactly identical, or they're not. And then, we can sort of put that to rest. Hey, they're the same, or hey, they're not. Because I think genetically through the DNA is probably the best way to tell if something is something or it isn't, if it's the same or not. Or you can't have two things genetically the same that are different, correct? Or can you?
Well, you can. So you can have. So think about piebald, for example, right? Not every piebald animal looks the same as every other piebald animal, in terms of where the spots are, how much of the animal is white versus how much is spotted, and similarly, you can have a gene that causes a major change in the color, but there could be all sorts of many other genes in that animal that have very more subtle effects. And so if you do a test for the thing that causes a major change, you'll know what category the animal would be in, but you might not know exactly what it's going to look like, subtle differences one way or the other, because you haven't tested for all that other stuff.
So it's not easy, cut and dry, easy answer. Like everything else in our industry, it's never easy. Right. You really got to put your work in and.
And what I think is going on sometimes with these allelic morphs is that it could be the same gene, but different mutations on the same gene. So I've read papers in which, for example, on a gene that causes an all-white phenotype, it'll produce red-eyed mice if the mutation is in this one part of the gene, but it'll produce a black-eyed mice, all-white mice, if the mutation is in a different part of the gene. So with a lot of these allelic morphs, that could be the answer. It could be the same gene, but on different parts. And so the most straightforward way to find the genetic basis for these yellow belly and asphalt would be to sequence ivories and super asphalts. Although you could put in a bunch of freeways, but the signal, as I was mentioning before, won't be as big, but you could still discover it.
Heather, have you received my box yet today?
No.
Then it should be tomorrow. You've got a whole bunch of super asphalt combos, monsoons, and you got probably 50, 60 sheds.
That's great, because I'm on summer break, so I don't have anything to do. So no, that's awesome.
We'll keep you busy. Don't worry. So
one thing I wanted to say though is, people have to recognize we are the infancy of this system.
Yes.
This has been done before. There was nothing, there, and there's just the 2 papers out now. So, and it's exciting. It's so exciting.
Yeah, and it's a good point because, I was watching the live that Hannah did, and everyone in the questions can you do this? When can we do this? When can I do this? How fast can I get this? Yeah, slow everybody. I'm messaging. I'm not so bad now, but before it was hey, Heather, what about this? Hey, what about this? It's oh my God, Billy again. What does he want now? No, Billy, we can't have it by next week. It's going to take it's Billy again. But, now it's and I always say, it's funny because most people, who know me, can I call you for a quick second? Make it quick, but my quick seconds are hours. It's like these podcasts, it's shut up, Billy, enough. But, no, I think it's great, and we are moving in the right direction. I remember when Heather sent over, Alan's, discovery, and it's just no way, oh my God, this is so incredible. it's a step in the right direction. And where this goes in 2, 3, 4, 5 years, know it's coming. Hannah is getting in this at a whole different approach, and other people can you're actually teaching people how to do this by the masses, which is great. So I think it'll come faster down the road.
In relation to your comment about things taking time, one of the things I've learned in this project in terms of talking to non-scientists about it is how much people who aren't scientists don't recognize how long science takes. So I put a post on Reddit, for example, saying, I'm collecting sheds for this project, send me some sheds. And I got an email 2 weeks later by someone saying, hey, I saw your post on Reddit about sheds, and I have a shed for my animal, but it was 2 weeks ago, so I'm not sure if you still need it. Maybe your project is already finished by now. And I thought, oh no, it is not finished. Yeah. Please send us your shed.
Yeah. No, and I thought it was great too. And for people that haven't watched the other live stream that you were on, what is the longest? Because I know, Heather, you must have some from me from 4 or 5 years ago. Can you just use that right away, or is there a process that you've got to put it somewhere and rejuvenate it and then extract it, or is it as long as it's dry? It has a. Fine.
I mean, remember, people are able to get DNA out of Neanderthal bones that have been buried in the ground for tens of thousands of years. DNA is an extremely stable molecule. That's not true of all molecules, but DNA is extremely stable.
Oh, so cool. So there is, so what are some things that can, you definitely don't want to do to the shed? Like, oil it up, obviously. Like, don't, shine it up or put lacquer on it. But, I mean, is it better to be sent dry? Because I remember you were saying, to Samson that, put it in a Ziploc and put a thing on and seal it, where I leave mine aired out and I put them in like a wax sort of sandwich bag and I send them into Heather.
That would work fine too. Yeah, I ask people to dry it before they put it in the bag. That would be fine. I just asked for Ziploc bags because I wanted to make sure that the sheds were separate from one another.
Yeah.
But as long as the sheds are separate and they're dry, that would be totally acceptable.
That's awesome.
They just isolated DNA from a million-year-old mammoth molar. So that's like the longest now that they've been able to, the oldest DNA that they've extracted from.
That's crazy. Science is mind-blowing. It really is. Like, oh man.
But
if it's dry and also in a dark area, because UV light also damages DNA. So as long as you don't have it out in the sun, but besides that.
Oh, see, there you go. Because you know what you said? Because here's the thing. You know how you say, we want it dry. Let me take this. And, most people live in a nice, especially summertime. Let me take them and, lay them out on my porch where the sun is to dry them off. That'll sort of damage them as well.
I bet that would probably still be fine. You might not want to leave that there for like a really long time under super high UV light, but I bet just drying it on your porch would be totally acceptable.
Okay.
Yeah, I mostly read about that actually in a book I read about Neanderthals from Svante Pablo or something like that. Yeah,
yep.
Yeah, where he said that the biggest enemies for ancient DNA was moisture and UV light, and so that's why you usually want dry samples, but that are away from the sun.
The samples I send are pretty good, eh? I've sent one or two that are probably suspect. It's like, Billy, man, what the hell?
We have found that the samples from Florida tend to be the most infested with insects of all the samples we receive. That's the pattern we've noticed.
Oh, wow. Insects in what, like mites?
Like, I don't know, like flies.
Parasites?
Well, I don't know what they are. What I can tell you is I'll get the shed in. I won't notice any insects in it. But if I just leave it in my office for a few weeks, there will be insects that hatch inside that bag a few weeks later that presumably were present as eggs or larvae when I got the shed, but I couldn't see them because they were so tiny.
Oh, crazy.
I assume that's because in Florida it's warm enough that maybe people keep their snakes outdoors, and so there's just more stuff there. I don't know.
Well, that is crazy. I mean, and here's the thing: we're science. You never know. I mean, wow. I mean, I wouldn't even have thought something like that. That's crazy. How in this just journey, you're learning more and more. And now, when you get a shed in from no matter what, is there a place where
I put
you have
it in the freezer.
Oh, so you have to do a certain thing. Okay.
I do that to kill any insects that might be there. That's the only reason I put it in the freezer.
Now with eggs and stuff, if you put it doesn't it just make the eggs go dormant, and then when you take it out of the freezer?
That would depend on the species.
Okay.
And I think stuff in Florida isn't really used to, hibernating for the winter for frost. So
Makes sense.
And especially even insects that do go dormant, they basically need signals from the environment telling them to be dormant. So they wouldn't necessarily just be able to survive freezer.
Our Canadian flies are much tougher, so we'd have to
Yes. So the stuff that lives in Canada is going to be really different.
See, this is so cool. I didn't even thought, like, something like that I would never have thought of. You're sending a shed in, and next thing you know, it's transporting eggs, or like, that's crazy. Like, it's amazing what you can discover, right? When you really get down to it.
Your fruit bowl, though, Billy, your bananas, all of a sudden they got fruit flies everywhere. Where'd they come from?
Crazy. See this? I mean,
Billy,
yep.
Just wondering, have you ever seen a piebald lizard, like a piebald gecko?
No. Please go ahead and say.
Okay, well, I'm actually gonna put something here on the chat about just one that's been discovered. And I'm asking because we teamed up with researchers from the University of Georgia, who I think a couple of years ago were the first ones to apply gene editing techniques to squamate reptiles, which include lizards and snakes, to sort of functionally validate this mutation that we found on their lizard. So they made this protocol and optimized it for anoles, brown anoles. And so we induced a mutation in the brown anoles and produced actually what look like piebald anoles. Just the thing is that it doesn't look exactly like a piebald snake. Yeah. There are some similarities to what this reptile that I found on the internet, and to the phenotypic. Yeah. So some of the differences between the anole and the snake are actually similarities with this lizard that I found here. So I was just wondering if you had ever come across, piebald lizards.
No, I know.
We might need a photo for the figure.
Yeah. So what happens is when you send it to Zoom, I'm broadcasting through OBS Streamlabs to YouTube. So I can see the link, but I can't get it. Austin, can you copy and paste it?
I found if you type into Google piebald crested gecko, it's the first thing that comes up for me. So that could be a way people could get to it easily.
Thank you, Hannah. Make my job easier.
Yeah, it's already on the link.
Okay.
Comments.
So Alan, it looks to me when I look at this animal that most of the areas that don't have color are in the limbs and the ventral region. Is that also what you saw with the anole?
Yes, and the snout, but it's just not pearly white. There is hypopigmentation, so that's the catch with ours. So yeah, I was kind of hoping it would be totally white.
What color are the patches in the anoles? When you say
it's kind of brownish, but very but you could see that there's patches. There's like darker brown and like a bit lighter brown. I don't know. Maybe Heather's also seen the photos. Maybe she wants to describe.
Interesting.
Isn't there a chameleon too?
Yeah, and also so I think this is something that of how the piebald phenotype is expressed in lizards. If you also look up piebald chameleon, it has some of the similar characteristics, which the arms are white and the snout is white.
That's the translucence, I believe.
Right.
A friend of mine here was breeding them out in Niagara region years ago. I don't know if he's still doing it, but he had quite a bit of translucence. What's that, Heather?
Get those sheds.
So I should reach out to see if he's still doing it. But he had like quite the breeding pairs and quite a bunch of it, and he's reproduced them and everything else. So, but no, that's super cool.
Kind of related to that, I was wondering what you thought about the role that CRISPR Cas9 might one day have in the ball python industry.
Yeah.
Because I think with the mixing of genotyping and CRISPR-Cas9, that would totally revolutionize things, right? With the genotyping, you remove the speculation about a potential genotype. And with the CRISPR-Cas9, you don't have to wait generations to hit a particular morph. You could have your own little technician there doing the surgeries. And from a normal, you could get perhaps a triple recessive. say, if you're
Correct.
lucky, right?
So for everybody out there that doesn't know, so what CRISPR and what you just probably said. So once you have, like, you have the sequencing for piebald. So once you have sequencing for, say piebald, clown, desert ghost, monsoon, you'll have all those because I've been sending you sheds, and, you know, we'll have those, plus what Hannah's, you know, team and what they've done. So with CRISPR, I can actually go take a normal ball python while she's, you know, before she ovulates, I guess, you'd have to inject what we make. I guess you make the sequences, or I don't know, you make it in a test tube, or I'm not really sure, but
Yeah.
you basically you take that in a syringe, inject it at the right time into a normal ball python egg, and if I have, so I can actually make one of those eggs come out whatever genes I want. So if I wanted a desert ghost, monsoon, clown pied, lavender, I can have that come out in a normal ball python's egg.
You'd be pretty lucky to have that all come out with CRISPR. It's in theory possible, but yes.
So how?
At least make one or two of those.
Yeah.
Right.
Yeah.
Okay, so you know what? Screw the rest. Just give me a bunch of monsoon DGs. I'll be happy. I'm already making monsoon pies, so hey. But now here's the thing. When you do that, do you think it's possible for when you're, I guess, creating that animal, is it possible, or has it been proven, that when you make that genetically modified egg that turns out to that animal that you sort of pre-programmed to make, can that reproduce and pass on those genetics?
Yeah, definitely. Yeah. And even more, if you find out that if you can induce a mutation, say if you know the spider gene, and you can induce that mutation in a nearby area, you might be able to reproduce the spider phenotype without the wobble. So that's another one of those benefits.
So you can correct some genetics.
For sure, yeah. Whether it's
Possibly.
the same, yeah.
Let's make spider great again. I had to, guys. Sorry. Oh man. Oh boy.
I have a few super chats.
Okay, go ahead.
Benevolent reptiles. How many sections of DNA strands are you looking at? Does each section mean that everything within that section is allelic?
Maybe this one's for Hannah.
So our approaches are different. So in Alan's approach, he's looking at the whole genome, the whole thing. In my approach, I'm making some guesses and looking specifically at those regions. The second part of the question was something about, can you read the second part of the question again?
Yep. Does each section mean that everything within the section is allelic?
Yeah, so by section, if we're referring to gene, which is a section of the genome, typically any mutations that happen within that section will be allelic. They don't necessarily result in an animal that looks exactly the same. They could result in an animal that looks a little different. So a good example would be toffee and candy versus albino. The toffee candy animals look a little different than albinos, but the phenotype would tend to be similar.
Now you can make a triple allelic animal, as long as the allelics are not the exact identical.
But there's only two chromosomes. They have two pairs of chromosomes, one from the mother, one from the father. So
three different locus, right? It could be a mutation on, say, on exon 1, mutation on exon 2, and mutation on exon 3. Right? That's how you could, and if those were all for morphs that were allelic. So what's like 3 morphs that are allelic?
Well,
so I guess
say black,
Lesser, Mojave,
black pastel, and Enchi.
Okay, so
chocolate spot nose.
So there are morphs that could behave like they're allelic because they're in genes that are close together, but the genes are actually different genes, and so it would be very hard to get to combine those genes together into the same animal. When geneticists talk about allelic, we typically mean something in the same gene. So in the case of like cinnamon and black pastel, my guess is that those are things that affect the same gene, and the Enchi is my guess, and maybe you guys know, Alan already know, but my guess is that something in a nearby gene.
linked genes.
Yes.
They travel together.
Yeah. It's kind of like how banana and sex determination travel together most of the time because they're really close together on the chromosome. So when you get one, you typically get the other one too.
Okay, that makes a little sense. So, yeah, it's just crazy what you know, getting this deep and figuring out why things and
Yeah,
how
well,
things
just
are.
Imagine how the early geneticists must have felt when they didn't know any of this. And they were just like studying animals and plants and trying to figure out what are these patterns that we see? Why do they behave this way? And they had to come up with all, this is before they even knew the structure of DNA. They were just trying to figure out what are these patterns? What are the rules here? What's going on?
Yeah, they probably had it like, I think we're onto something. And then, oh, nope, throw that all. Let's start from scratch. Here you go.
Yeah.
And then it's like, remember that stuff we did like months ago? Bring it back, bring it
Yes,
back. And
yes.
yeah, trying to figure out that thing. And I know it's definitely, you know, being in that and trying to figure it out. I mean, it's just, it's a mystery, and it's something that, you know, you probably go to sleep dreaming of it. I got to get back to the lab. I got to figure this out. Or, oh, maybe I didn't do this. I mean, it's just, it's so great. It's like us creating, you know, different morphs. We're doing the sort of the same thing, but not as amazing as you guys. Like, you know, such a different level with hardly any understanding. It's like, take this and this, put it together and do this. But we try to do the same thing. Like, let's take this and hopefully it looks like this because this should add some pattern. Oh, it didn't. Okay, well, let's take this away and add this, and there you go. And we start sort of, you know, altering and trying to figure different things out to achieve that goal of ours. But I mean, it's just amazing how where you guys are at now, and especially, you know, where we can go, especially with what the data and everything that you guys have already come across now. Moving forward is going to be a lot easier than, you know, the last 4 years to where you are now. Or I hope it is anyways.
And color morphs have been since the beginning, right? Like since Mendel that discovered particulate inheritance, right? Pretty much meaning that the genetic basis for traits were in pieces or sections of DNA. And yeah, so we're pretty much just following on the tradition of Mendel, I think.
Here's another super chat, which is awesome, from Colorado Cold Bloods. Take it away, Austin.
Will we be able to determine the difference between the different lines of Xanthic, MJ versus TSK versus VPI, etc., since we know they are not compatible?
Great question. So definitely, it would definitely look differently, correct? On the
Yeah, we just need the sheds. We need samples of the sheds. Yeah. Yeah.
Cool. See? So send them in. Your info too, I'll get, and I'll put it down below. I didn't have the info to where to send, or a link where I can send it so people can get a hold of you, Hannah, and send in the stuff in the U.S. And that's another thing too, is you know, where what we have to deal with is because these are a CITES animal. You know, the same way we have to ship our animals and get the proper paperwork, and I definitely, you know, suggest you do that. Some people, I can just cross the border with it. And like, don't take a chance. I mean, there's a reason why these rules are in play. Please follow by them. You know, if you can't wait, you know, 2-3 weeks and spend $100 or $200 to ship an animal, you know, you shouldn't be acquiring it, especially across borders. So do it the right way, please. But even with sheds, because they come from these animals, we still have to abide by the same rules. So we're going to look into that. We do have some ways to do it, but in the future, I'm looking into that as well. So, you know, because I know there's a bunch of people in the States that I want to work with, and we do have a way. But in you know, Hannah's case, everyone in the U.S., which is such a bigger gene pool with the amount of keepers, can, you know, gene pool, anyways, you can send the sheds into her. But we have a smaller base here, but I am going to reach out to some other Canadians as well as some other stuff, and even maybe Europe, and see if I can import different mutations from different keepers, from different lineage, et cetera, et cetera. And do you think, either of you, Hannah or Alan, do you think that different lineages are going to show difference of the same animal?
Different lineages? Yeah, so I think you're meaning like has more than one mutation arose multiple times that explains a particular morph. That could happen, right? And I think Yeah, maybe, perhaps, but I think for the co-dominant ones where that might be more selected against the nature, the recessive ones might be able to kind of sneak by, right? Because you need kind of both copies before that animal would be kind of at a disadvantage in nature. So yeah, that's a possibility, I think.
Awesome. Yeah, because I mean, I know that pretty much the majority of the sheds that are coming to you guys are through myself with my genetics, my lineage, stuff like that. I just want to see if, other stuff that comes in that's not related, is if it's going to show, a different, or is it still going to be, very similar?
So what would be interesting is when these original individuals were brought into the hobby from Africa, were they coming from the same population, or were they coming from different populations? Because if they're from the same population, you think that probably was the same mutation. But, we don't know. Some of that stuff is so far in history,
Yeah.
but the genetics will tell. We just have to take the time to look for it.
Yeah, it would be interesting to get a sample from one of the original piebalds and check it for the mutation we found, for example.
Now, do you think mutations would change by breeding over years, like to a degree, to where it's like, you find the original pied that came in 35 years ago to a piebald now, that's a direct relative way down the line, is still going to be so remotely similar that or is it going to be, you don't know, obviously, because you haven't looked, but, with what you're understanding and stuff, would it be the similar, or would it be like, holy God, like it's changed so much over 35 years by breeding?
The piebald mutation will, if you keep breeding it, it'll remain there, right? Millions of years of it, theoretically. But mutations can arise, right, as you're breeding. So in your line of piebalds, you could have a mutation that arises that it doesn't affect the piebald per se, the Mendelian phenotype, but as Anna was saying, it affects these other more subtle, more quantitative changes, right? How much is it a low white, or is it a high white? Is it does it brown out as it ages? These sort of things could arise but because random mutations arise.
Now here's a question. It might be way out of left field, but the puzzle gene. Now, I heard the puzzle gene was never a gene because the original parents that bred and this animal came out, it's like, what the hell is this thing? Those animals were bred year after year after year and never made the same result. I think they called it a spontaneous. Can you breed two animals and during incubation or whatever, just all of a sudden a new gene pops out? Is that realistic? Yeah. Is that something that can happen? Or because that's what they say with puzzle.
That's why we're not swimming in the sea anymore.
Sorry?
Right? That's why we're not swimming in the ocean anymore. That's how evolution works, is you get these spontaneous mutations and they have a change in the phenotype. If it is not beneficial, that line dies out. If it's beneficial, you end up, giving podcasts many millions of years later.
Your mic's off, Hannah.
Doesn't say muted.
Yeah, it doesn't. I did for a second.
Okay, let me try again. Are you
Nope,
hearing me?
We can hear you now. Yep.
Okay. So if a mutation is recessive, then it can just literally pop out, because in previous generations it was always hidden by the fact that there was only one copy of it in the animal. So if you all of a sudden see something that pops out, it could be just a recessive color morph that nobody ever saw before because it was never present in two copies.
So how would a mom and a dad pull out an animal, and you breed the same pair year after year after year after year after year and never produce?
Yeah, okay. So that's a fair point. So yes, that would likely be a different situation. I have forgotten that you introduced that into the scenario. So
yeah, because to me it's like, what? You can just have a gene just spontaneously pop out? That's nuts.
Well, okay. So technically, there are new mutations that can happen each time an egg is created, and each time a sperm is created, there are new mutations that sperm has and that egg has that were not in the parents. So for example, each one of us received about 20 new mutations from the egg that created us, and about 20 to 40 new mutations from the sperm that created us. older fathers tend to give more mutations than younger fathers. So it could be a new mutation that happened specifically in the sperm or the egg that created that animal, and therefore it wouldn't have been present in all the siblings because they were made using different eggs and different sperm.
See, that's it's science and genetics, and I mean, it's just amazing. Like it is. It's like I can see your attraction to it because it's like you want to find out answers and you want to, I mean, it's just amazing. It blows me away. And I just find that we're going to start finding a lot more things down the road through science and your work that we can apply. And what about like birth defects and like two-headed snakes and twins? How does that get into the mix? Like, will you be able to sort of find out why that happens, or is there a reason behind it? And or is that's totally away from your scope of work?
I think that's harder because you'd have to collect a lot of animals. You first have to have evidence that was happening because of a genetic reason. Those sorts of developmental issues can also be essentially non-genetic. They can be something that happened during development that wasn't necessarily caused by the genes.
The incubation or something like that?
Yes, or just things that you can't even control, or things you wouldn't even know about. So you would have to, I think, first collect a lot of animals that had the same type of defect over and over again and had evidence that it was genetic, and then you could figure it out. But just a one-off situation, that would be hard to identify. Even figuring out whether it was or wasn't genetic would be difficult with just a single individual.
Okay, I guess there's something going on in the chat. Okay, so let me, 1 second. How do I do this? Guess I gotta do it from my phone. Hang on. We always get that odd person in the chat that just...
I'll tell a little story of a clutch that I just had. I had a female who had 14 eggs. Unfortunately, she only laid 12. She actually did end up laying the 13th a day later, and I kept that egg, and it actually developed. The 14th had to be surgically removed, but that 13th egg was severely kinked. I ended up cutting it and then euthanizing it. But I mean, it could be a complete coincidence, but that was the one egg, and all the others are fine, that was retained for an extra day. The temperatures may have been different, and so that would not be a genetic mutation. That was something that happened because of the environment.
What about parthenogenesis? What's, basically that is a female reproducing on her own. Why does that occur? And the funny thing is, the first 10 or 12 years when I was doing this, it was like, what's that? Oh, that can happen? Now it's like happening. I'm getting it 3 times, 4 times a year, but I'm also breeding a lot more. And I think you're starting to see a lot more of it happen because there's so many breedings compared to 10, 12 years ago.
And you might be just on the lookout for it more now too.
Oh yeah, that's another good thing. Yes. Now, what? How does that, explain to me? I mean, I want to know, maybe other people do, and I know it's off the genetics, but I mean, no, I guess it is sort of genetics related, but how does that, it's just amazing how a female can reproduce on her own, and I know I have a few, I have some, and they're actually still doing well, but a lot of people say that they don't really last long, they can't breed. I know there's other people doing some studies, and I had someone reach out that was actually studying this a few years ago when I had my first one, maybe about 3 or 4 years back. And they're like, yeah, we have some, most of them end up in our freezer. But most of the ones I have are still doing well and thriving. What's, the whole thing about the parthenogenesis of how they can duplicate their genetics on their own without even a male, which is just another thing that's crazy. It's just what? That can happen? It's just mind-blowing.
So I teach this, so I get super excited about it. There's actually populations of animals, reptiles, the whip-tailed lizard that's in the southwest US, that there are only females. Oh. There are rotifers, there are fish that do this. There are only females. And they actually still, especially the whip-tailed lizards, they still do the courtship. So they evolve from populations with 2 sexes, but basically they ovulate the egg and then it starts undergoing mitosis and creates a new individual that's going to be a clone of the female they came from.
It's nuts.
Yeah, and since the female has both genomes, right? They have the mitochondrial genome and the nuclear genome. They don't really need, the other copy. They don't need the male much. So yeah, you could do that just with the one copy of the genome and the mitochondrial. Although exactly the molecular triggers that cause parthenogenesis, I'm not too sure. Like for example, in pythons.
So nuts. It's amazing what we're going to find out moving forward too through all this, and put a lot of, I guess, theories at rest, or solidify them. But I think it's really awesome. Now, I don't know, where do we go from here? I'm just trying to think. I'm trying to read the comments and trying to get, I guess those other two were blocked already, which is great.
I think in science, though, that the more we learn, the more questions we have. And that's why, pooling different resources, different individuals with their knowledge. I mean, it was kind of serendipitous that, we found Hannah and Chiron, and actually that we found Alan and Chiron found Hannah. Like, it was this serendipitous event where you have these Python enthusiasts that find the geneticists that can do the work. And I think that this will spur a lot more interest. And yeah.
I've got to ask Karan a question. Are you a breeder, or have you actually successfully bred, or it's, you're in the works?
So I have successfully bred, and actually some of the samples that originally helped us to identify the mutation in albino were some of my,
awesome.
Came from one of my clutches. And so right now I have a Tofino, expected Tofino clutch to kind of come out. And so we're kind of going with that idea of identifying these mutations, being able to genotype animals, because as you know, because they're allelic, some of those babies are going to come out either het albino or het Toffee, and I'm not going to know which one they are. And so this is one of the things I want to say that's exciting about this project, from an educator standpoint, particularly science educator standpoint, is being able to bring people who may not be interested in a hobby in. At the same time, sometimes science class can get kind of stale. You bring in something that has some relevance, right? Like, it's like we're trying to do this work not because we already know the answer, but we're trying to help out this breeder, this local breeder. They want to know what particular morph they have. And so the answers that we come up with are actually going to something other than a professor grading a paper or a teacher grading a paper.
Which I think is amazing, yes.
Yeah, for me, it's like, this is an opportunity for us to kind of promote the hobby and at the same time engage in a way that can just transform science education. So, I'm over the moon with this. But I've got to, hopefully this clutch will pan out, and then Hannah will have samples for her students in the fall, and I'll have some answers, and I can sell these guys and girls as either het albino or het Toffee.
Oh, so you don't have to put the labels het Toffee / het albino.
Het albino,
Your label,
exactly.
you'll be saving thousands of dollars just on label makers, on labels, and it's crazy. Now, have you tested that animal? Are you sure it's Toffino? Are you sure? Or is it Toffee?
We're pretty sure it's Toffee. One thing that's that we're still trying to, so I've actually got I've got a female Candino and a male Toffino. And so one of the things I want to do, as Hannah was talking about earlier, some of our preliminary data shows that they're essentially the same allele. And so it's just a matter of, what name are we going to go with? But we've got this, I've got this Candino that I'm hoping to breed next season and so forth. That'll be a project and opportunity for students to be able to kind of put more data out there to put that side of the controversy to rest. It's actually the same thing, Candy.
You're gonna upset a few people.
I know.
But you know what? You can't fuck with science. I mean, you know what? Here you go. Here, read it and weep. Here's the facts. You do what you want with them, but the fact is that here you go. So Your mic, Hannah. There you go.
Why, when you hear me now?
Yep.
Finding the history of these morphs has been somewhat difficult. So for example, the toffee and candy, when I started posting about it on Facebook, trying to recruit sheds, someone would say, oh, these are the same. They were siblings that came out of the same family, so they're the same. And then somebody else said, oh, they were found under the same bush in Africa, so they're probably the same. And like, okay, both of those stories probably aren't, like, they can't both be true. So is even one of them true? Who knows?
Yep. No, it's crazy. But like you were saying, making science exciting. Like, to me, I've never been a science person. Like, I've always I'm a mathematics guy. So science and history was never my thing. But, if with this, like, oh my God, like, I would love to learn science this way because it's, I have a passion for the snakes. And I think it's great because the way people, and you have a new way to learn it and do what you would normally do with ball pythons, it makes it exciting for many people. And I do think that some people, while they do this, will start being introduced to these animals or looking into them and looking at the colors and go, wow, and vice versa. You're educating people and getting people to want to be, educated, and other people that are educated that want to get into the, the snake or learn about it. I mean, it's a win-win.
And the other thing too that I'll say, is, I mean, you've probably seen it, Billy, where you've got a bunch of people that have a YouTube video where they're trying to explain banana genetics, or they're trying to explain some other type of genetics, and they kind of miss the mark a little bit because they don't quite understand it. This is our opportunity to serve as ambassadors to kind of connect the two, so people actually understand the genetics, not just from the ball python standpoint, but just from a genetic disorder standpoint, or any of the other things that as educators, we're trying to help our students understand and the population understand. So
yeah, when you figure it out, we just need the way to transform it so that all of us, people that aren't science majors understands it. We need a translator. But I mean, it's definitely amazing that, where we're at and where we can go with it. I just think it's great for the industry. I really do. And never mind finding out all these other things that were just like, like Hannah said, there's all these different stories. Like, which one's true? You never know. Well, hey, here's fact, here's science. Who cares what the stories were? Here's the results. Do with what you want with them. I hear a fan in the background. Any more super chats?
I imagine, just as there's been like a huge push in academia for sort of open science and having like open data, making your data available, I can see something like that happen in the Python industry too, right? Where maybe you include like what in genetics is called like a FASTA file, which has like the genetic sequence of a particular animal. You can say here, this animal, this is the sequence for this animal at the piebald locus, here's the mutation, right? Of course, I guess maybe people could make up a sequence and say like that's theirs too, but yeah, I guess you would need a third party organization sort of to certify that it is what the sequence says it is.
Yeah, so now moving back towards that, so I know Hannah's getting a lot of sheds in for her students for the fall semester, and even though someone sends you a shed, they're not going to get the results back in a week or two. It's going to be, a while after processing and stuff like that. Now, you're going to be doing this for a while, and there's so many, genes to crack or find out where they are. How realistic to, Alan or Hannah, do you think that this would be where, we have, 100 different genes where people could actually send sheds in? And like, are we 3-4 years away? It's something that's undetermined, but with where we see things now, because, I know when this opens up, people are just going to say, hey, here's sheds, just let me know what everything is and stuff like that. I mean, is it going to be cost effective to do this? Is it something that it can be turned around in a timely manner? And when do you think we'll be at that point where, we either, have a commercial industry for this that people oversee and govern, with credibility of what these animals are and doing it right? compared to, like you said, it's probably not feasible that people will be doing this in their home. So there's got to be some sort of way, because you don't want everyone in the ball python world, Hannah, to be sending you sheds, because you'd have like dump trucks coming every week. Here you go, Hannah. Beep beep. What the heck? Open up the freezer. You're gonna need a bigger freezer. Your mic again.
The turnaround would be pretty fast.
Okay.
Whether it's technologies that I'm using or the technology that Alan described, potentially creating, where there was like a chip that had a simultaneous test for a lot of different genes. Turnaround would be like within a couple of weeks, assuming that the test was started on the day that the person received it.
Okay.
But of course, whether that they would start the test on the day they receive it, it depends on how busy they are, what the company is, and so on.
So
in terms of cost, so currently the supplies to just test for one thing are very inexpensive if you're not paying a human to do this. So if I'm having a student do it, it would be, really cheap, like $20 or less. The chip technology, I think, Alan, would be more expensive. I think on the order of like a few $100s for a chip for a bunch of different morphs. So I think those are the costs that I can say right now. Alan, you want to add more?
Yeah, there's different chip formats, and just what I've been talking to the McGill Genome Center is that you get deals the more samples you send in at once. So there's this one format where you can genotype at around like 24 SNPs. But if you send in 1,000 individuals, you get that down to, I can't remember the price they gave us. This was a while ago, but it was under $100, definitely under $50 per individual. But you have to send in like about 1,000 of them at once.
Yeah, so your turnaround there might be slower because you'd have to wait till you had 1,000 animals to do all at once.
Yeah. And in terms of somebody sending in a shed, if it's to genotype a mutation that we already know, the turnaround time, as Hannah mentioned, won't be too much. But if they're sending in a shed to discover the genetic basis of a particular morph, that's going to take a little bit longer.
So if we had, say, 50 morphs that you knew the location and the sequence for, I send a shed in, you send it in, you can come back and say, hey, without any information, you can say, hey, this shed that you sent me, it's a super pastel, NG, yellow belly, pied clown, 50% het albino.
Yeah, but it wouldn't be 50% het.
Oh, sorry, not 50%. Jeez. Thank you. I mean, duh. See, I need a
That's
coffee.
what we're trying to move away from.
Yeah, see? Yeah, exactly. See, Billy? That's why we need these, awesome people. Like, I love how you're just correcting me right off the bat. I love it, see? Awesome.
So,
and you guys know it. So it's crazy when I get so excited when people, especially new, because this isn't something that you've been in or, you've studied ball pythons, and here you are knowing, 50% het percent, like, you're starting to know the lingo that we say and the genetics. I think it's so cool. And especially kids. Kids are coming in now and they get the, understand, oh no, that's recessive. You breed that. It's 1 in 4 chance to hit the visual. I'm like, it's so awesome. So pretty cool.
So if we had 50 sheds, now that we have the protocol ready, now that things are opening up back in Canada, post-COVID, I think we can have a turnaround time of weeks, I think. And so, we need 50 individuals of that particular morph, but we need to compare those to another pool. So what we want to do is, what we did in the piebald was compared 50 individuals of piebald with 50 that don't, with 50 individuals that don't have piebald, but have an assortment of different mutations. So to speed up the process, what we want to do now, is we want to find out the mutation for clown and pastel, is compare a pool of 50 clowns that don't have any pastel with a pool of 50 super pastels that don't have any clowns. If we have pools like that, it could be quickly.
Oh, no, it's coming. We got it. I mean, you just leave that to me. I got it. Don't worry. Yeah. I mean, I think this is something that the community definitely want to help out in and get to do this. And I think down the road, it's regardless of where it goes, it's something that's going to be beneficial to our industry. It'll help breeders. It'll help purchasing, selling. It's just amazing. And I think, with Hannah's undergraduates or their students, you're going to have a lot more people going, wow, man, like these animals are cool. I might want to get one or two. And then they're future breeders. And then they're going to have a podcast here and say, hey, I have my original teacher, Hannah, on here. And I mean, it's so awesome. Like, you know, just keeping this industry going and going in a proper direction. Like, I think this is it's super cool and fascinating. And, you know, I thank you guys for all your work and everything that you're doing. And, you know, I mean, I want to help in any way. And I just think it's definitely needed because, I mean, you know, what's what, this and that, this is this, how it works. And half of us are wrong. And it's like, oh, hello, I didn't know that. We assume with the best of our knowledge where you guys can put it to rest. And I think that's key, you know, and moving forward, especially because, you know, there's going to be more genes popping up. How did that come out? How does it work? Here you go. But I think moving forward, especially 5 years from now, quad het to quad het is the norm. Like, it's going to be like, you know, what you're doing. I mean, if you're not doing quad het to quad het or something like visual triple het to visual triple het, you're missing the boat. You know, in 5 years, if you're not doing that, I mean, like, you know, that's where we're going. And we will definitely need this information and stuff in order to make it a little easier because no matter what, you know, we're shooting quad het to quad het to hit that animal 132 or 160 or whatever it is. We can't change that. That's still going to be there. But knowing that I have a true quad het to a true quad het, that's the key. You know, because if you're shooting a quad het to a triple het and you're trying to hit the quad, it's never going to happen. I don't care how many times you breed it. So knowing that, I think, is incredible. So right now, how many genes do we know? Pied, albino, toffino, or sorry, well, toffino, but toffee, candy, ultramel?
Yes, ultramel.
Yeah, it's the best gene in the world. So you have to do ultramel.
Albino too.
And lavender albino.
And lav. I had to plug ultramel, baby. So see how Austin just sort of woke up? Ultramel? Zach's going to pop up here. Like, someone say ultramel? Zach Nava. So what do we have? Let's do this again. Pied, ultramel. Do we have caramel or monarch?
So no, but only because I don't have any samples for those. So if someone could send me some samples, please. Those I have some good ideas for what genes those are, and I just need to receive samples.
Awesome. So we're looking at what, seven genes right now?
Well, I would consider toffee, candy, and albino the same gene, but you can consider them different morphs.
Okay.
Yeah, four for sure. Basically, four genes, potentially more than four morphs if you're considering allelic things to be different, but 4 genes.
Awesome. That's super cool. You know, just think, it's just like you know, like Heather said, this is infancy. Like, it's crazy because now, you know, we can talk in a year from now. It's like, yeah, we're 44 now, you know, but we're on the way. We're sequencing 65 others. You never know, right? But, you know, it's just like the foot's in the door now. And now we just got to keep moving forward. And I think it's great how people are learning. You know, it's being used to educate people and bring awareness. And I mean, it's like a win-win all around for you know, not just keepers and breeders. It's for you know, the way you're educating people and teaching. And, you know, it actually might make people go, hey, man, I want to take this class now. It's pretty damn cool, you know? Back in the day of just dissecting a frog or something, like, you know, hey, you know, a frog or a pig or a worm. Now it's like, we can do stuff with ball python sheds. What? Oh, cool.
Yeah, or like as I was telling Heather, like before you would do this sort of analysis with like cytochrome oxidase, these sort of, boring sounding chemical. Now with, you know, with this, I would be far more excited. So I think it's totally cool what Hannah and Chiron are doing.
Yeah, no,
I think
definitely.
another thing that we're going to see is like there was this lull when scientists weren't necessarily looking into ball pythons, and now that we've kind of opened the door and shown that, hey, this is a resource from a scientist standpoint. Hey, this is a resource you may want to start looking into. It's just like with everything. Once people see that, hey, that's an avenue, there are going to be more groups popping up that are studying this. So it's like you said, Billy, we've got our foot in the door. Now that the foot is in the door, we're going to see like this waterfall effect where more and more researchers are getting into it.
Well, I think that's great.
Yeah.
Yeah, especially because look at the amount of genes that are out there. Look at the pool. It's not like you're working with 6 different animals and 6 different genes. I mean, there's hundreds of thousands, if not millions of ball pythons. And look how many genetics, look how many combos. I mean, it's, we're not even, we can never, it's infinite. So, I mean, I can see where it attracts, that attention. And I think it's great. And, I'm glad, you guys here have, opened up that door. So I'm excited to see where it's going. And I'm sure you guys are as well.
Yes, very excited.
Why are you smiling, Heather? She's all smiling.
Send in those sheds.
No, definitely. And, everybody, I'm going to get that link from you, and then I'll repost it on here. Just the way to send it in, your Facebook link. And I know you're just working on Instagram now, right? You're just getting into that social media. And the only reason why you got into it, I'll tell the story about how they thought you were a bot. That was hilarious. So.
Yeah, I'll send you an email after this with some links that you can post.
Okay. Tell the story about how, you're never on media and you got onto social media because of this.
I didn't know anything about Facebook or anything. I decided that it would be a good platform for recruiting sheds. So I made a Facebook profile for myself, Ball Python Genetics. And then I found there were some groups. So I decided, well, I'll just join all the groups about ball pythons. There's lots of them, and then I'll be able to post to them. And I joined so many groups that Facebook said, you've joined too many groups. You're clearly a robot. You're not allowed to join any more groups for 10 days or whatever.
She went to Facebook jail the very first time she goes on. Oh, Facebook. See? That's why Instagram's so much better. Oh.
Yeah, my students have been helping me with the social media part. They explained, how all this works and how to use the messenger and how, yes, there's a lot for me to learn, actually.
Well, you're probably sitting there going like, see, I knew there was a reason why I didn't want to go on Facebook in the first place. Yes. You were smart,
Yes.
You should have stayed away, Hannah. You should have stayed away. Yeah.
But that's the reason why this project keeps on giving, because it's like we're finding more and more ways to connect with our students. So it's not just learning about genetics in a way that is more exciting in the old way, but now they're getting opportunities to show off what they know.
Yes.
And that's the thing so I teach teachers, and that's one of the things that I emphasize for these future teachers. It's like, don't downplay what your students bring to the table. In fact, use it as a resource, and then that makes the whole experience that much richer.
100%. And the funny thing is I've learned so much just having other breeders on my podcast. And, one of the ones I'll shout out is Earl from Lone Star Reptiles. He was on as a guest, and he's like, yeah, somebody asked, because we'll answer some questions from the chat before we end. And I also got to give out the shout outs of the week. I'll do that after. But what happens is, someone's like, hey, how do you you guys have any tricks? Because you got so much knowledge of how to get your ball python hatchlings to eat, It's always the same thing here in this. And Earl goes, yeah, well, there's another one. Try to get some vanilla extract. It has to be pure vanilla extract. You just put a little drop on the back of the rat. And I'm like, what? And I was like, what? I've never heard that. You're kidding me, right? He's like, no, try it. Sure enough, Catherine, who works with us, one of Team MC, she goes, brings some over, puts it on, and these things are eating. Like what? So it's amazing what you can learn just by speaking to other people, whether they're new, and what's funny, I've been doing this for so long, and some people that are new will ask me a question that in 20 years I've never even thought, like, what? I don't even know. I mean, I've never thought of it because from their perspective, or question or something that they don't understand, I just walked over it. I never even thought of it. I'm like, you know what? I don't even know that. That's a great question. And, it's coming from someone that's been in this for 6 months, and I've been doing this for 20 years, and I've never even thought of that, or I don't even know how to answer it. And it makes me go and look and dig and go, hey, man, here, I'll get you an answer. And if you find something, and I connect with them, and I'm learning from somebody because they've encountered something, and we all will. We're never going to always have the same path, encounter the same things. And I think that's another thing that's great, is listening and opening up your minds and working with other people, no matter how, you're a teacher. I can't learn anything from my students. If you have that type of mentality, you're losing a lot, I think. And I think it's great for being a teacher and connecting that way. And like Chiron said, is it's a great way to connect and make them feel that they're, you know, especially when you can take someone to teach, hey, I'm teaching my teacher how to use Instagram or social media. I'm sure that makes them feel amazing, right? Because it's like, what?
Yeah. A student had to explain to me what the word canceled means, and that it doesn't mean that like an event was canceled necessarily. It can have other meanings also.
You know, and I can't keep up with them all. I mean, every 6 months there's something new. You know, you still use that word? That's so old. That's not even, you can't even say that's so old anymore. Just, I mean, that's really, really old. There's a new saying. You know, it's crazy. But I mean, it's good to always be learning.
Yeah, I just thought of something that we hadn't discussed, like the possibility of the genetics techniques that the both of us are using and what we can gain from it. You know, another thing that the industry can gain from it is definite paternity, because we've got that situation. I've got a particular situation that I'm going to have Hannah testing to ensure that the father who I think is the father is the actual father, because I had a couple of unexpected hatchlings come out that just don't make sense at all. And so using some of the techniques that Hannah and I use to test paternity, that's going to help the industry as well.
Oh yeah, because there's retained sperm, you know, Parthenogenesis. Mm-hmm. Now here's a question. It might be can you have? So I know we've done it. Well, I mean, we weren't trying to do it, but that gentleman named Austin that's looking at his phone, it's really quiet. He bred one female to multiple females, actually 3, and we actually had 3 daddies in the clutch. So 3 daddies. So 1 egg was from one dad, I think 3, and then 4 from, but they were all clearly, you know, because there was no, each male was different, that we could not confuse the 2. So we definitely had a clutch where there was 3 males out of 8 eggs. Now, can you have, say, an egg or 2 that's a Parthenogenesis egg? I mean, or does the whole clutch have to be Partho? Can you have like, you know, 1 or 2 eggs being Parthenogenesis and then other ones through that? And then there's also, you know, going back to, you know, your thing, you can actually have retained sperm. So you can have a whole bunch of different ways that a hatchling can come about. But can Parthenogenesis, does everything have to be, because what I've noticed is everything's always been female, the whole clutch. I think this is Heather's question. So can I have 1 baby that's a Partho and the rest came from a male?
So you realize that we're just throwing out hypotheses, right? Because yes, but I think it really depends on what the trigger is for parthenogenesis, and I'm not sure we don't know what it is yet in ball pythons. Now, in terms of having, you know, multiple fathers, I'm just going to say that all those sperms swim really well, and that's not unusual, I don't think. But I think we have to know what the signal is for parthenogenesis to be able to answer that question.
Okay.
And how long spiders, or how long pythons could hold sperm, right? Is it just a few months, or is it an entire breeding season?
And we'll have the answer from you guys, what, in end of the month?
Next week.
Next week.
I think it's not out of the question that you could have, in theory, knowing the biology, it's not out of the question that you could have a clutch of mixed parthenogenesis and not, but that doesn't mean that happens very commonly.
Yeah, because here's an example why this actually came up, and I think this is what, you know, another situation where this is going to be super helpful. I have a breeding loan, so I have an animal in Europe. It's actually lightning pied, so MJ exanthic piebald, and you know he's breeding some double hets and some stuff. It was bred to a mahogany het albino. So basically, we're going to get mahogany, you know, double hets, 50% het albinos. 1 female came out, and it was a mahogany albino. So he asked me, it's like, hey, is this possible het albino? And I'm like, could be. I don't know. I mean, I don't think so, but everything's possible. And I'm like, what about it being a Partho? Maybe it's a Partho because, and he's like, no, because if it was a Partho, then it would have been a Summa. Because isn't it like mahogany, and then het to het? So it would have to be a Summa albino. You couldn't, oh no, so it could be a mahogany albino.
So the mother was mahogany.
Het albino.
Het albino. I don't think that you would have to assume that a parthenogenic offspring would be Summa.
So isn't it like double mahogany and then double het? pied? I mean, het albino, so you'd have to have an albino, and then het mahogany. I mean, mahogany would be summa. Isn't it like
where that allele was, which chromosome?
Yeah.
If they were on opposite chromosomes, yeah.
So I could hit a mahogany albino from a Parthenogenesis without hitting a summa.
Yeah.
Yeah.
Yes.
Okay, but I mean I'm stumped, and I'm like, no, maybe it's a Partho because it's a female. Did you get? And he goes, yeah, I got a bunch of other males and females, but this one's a female. The only albino mahogany in the clutch. Is it 50%? I'm like, I don't know, and that's why I threw that out. He goes, no, because if it was, it's usually the whole clutch. That's why I'm asking. I'm learning myself, and taking advantage of your knowledge.
Throw out there that keep in mind how difficult it would be to recognize a parthenogenic animal amongst a big clutch that wasn't parthenogenic. You'd only be able to recognize it in situations like you just suggested, where it was the genetics were such that it could not have come from the father. So that's just something to keep in mind, that it might be that if mixed clutches are more common, they might be like under recognized just simply because they'd be harder to notice than a clutch that was all female.
Right, like something like this, I would just basically go to find out how that animal existed, test the DNA of the male. Nope, it's just a pied MJ exanthic, no het albino, and then the only way that could be done, because it wasn't bred before, Partho.
Yeah.
So it's going to be so helpful down the road in so many ways. But you know what? I'm actually hoping it is het albino, because now we have an MJ exanthic pied het albino. That'd be cool too. So, or you have to breed that to another albino, and hope that comes out, or you just, oh man, I didn't prove out. Oh, it was a Partho. Oh, I just wasted another albino. Now I got these triple hets. Damn it. So, next year, but you learn what it is. So it's going to be so. And a lot of people say, aren't you going to take the fun out of learning and trying to discover this stuff? Yes and no. I mean, it's like, maybe new people, but I've been doing this for 20 years, that I've have animals that I've had to prove out. I still got 66% hets. I got visual hets, 50% hets, 66% hets, possible hets. It's, I feel like I deserve a little bit of a cheat. You know what I mean? So, and even with you want to know, do I really want to sell them as 50% or 100% het albino or het toffees? Do I want to price them that way? Or do I want to know what they are, sell them or keep them? I think the way the industry is now, there's so many things to chase and try and go after, where 20 years ago it was like you had 7 genes, it was fun to experiment and try to hit stuff. Where now it's like there's so much stuff that we want to hit, we don't have time to chase or, try to hit a 7-year project with, one of the animals that doesn't have one of the genetics that you're looking for. So it's neat. You're looking weird, Austin. What is it?
We got a couple of super chats.
Then interrupt. That way you save me from talking so much.
We got benevolent reptiles. How relevant is genetic studies in reptiles if somebody want to study piebald or albino in mammals?
Alan, do you want to take this?
Well, I think it is relevant, because a lot of the genes that cause piebaldism in invertebrates are shared across very many different vertebrates. For example, in humans, the same genes cause albinism from fish all the way to humans. So yeah, definitely discoveries that are made in ball pythons can be transferred over to studies in humans, including things that are like associated with like these pleiotropic effects. For example, the wobble, kinks, certain mutations that cause pigmentation disorders in humans. You also see these other secondary traits. And so you could use, for example, a ball python as a model to learn about these syndromes that are associated with pigmentation in humans.
Interesting.
We got another one from Forest Pythons, Bateman. It says, right now, are you guys looking for more single gene sheds or animals with multi-genes?
Both, right?
Yes, Everything is useful.
Yes.
Including normals are also useful.
What would you use the normal for? Just to use in say like you want 50 with piebald, one of those things where 50 without?
Yes, that's exactly right. And in some ways, normals are, I guess in my opinion, they're almost the most useful because they can be used as a comparison to any morph. And they also tend to be the most genetically diverse because they aren't something that has maybe been bred from a line where the parents were related and the grandparents were related. So you're getting some additional genetic diversity, which allows you to rule out certain mutations as causing or not causing certain color marks.
Yeah, that makes total sense. Like, you think about it. Like, wow. Hey, see that, guys? Normals are useful. Now people are going to, like, you go to Morph Market right now, and normal sales are through the roof. Everyone's $5,000 for a normal. What the hell? What's going on? Oh, you didn't see Billy's podcast? Wow. Normals. Oh, my God. Oh, boy. Only Justin Kabilka can do that. Come on now. You know?
Wild type, Billy. Wild type.
Wild type. I think we have another one, too. Last one.
William Philippik. And he says, Dr. Warren Booth does a lot with Parthenogenesis and reptiles. He would be the guy to talk to.
Yeah, I was on a podcast with Chris Eaton from Snakes and the Fat Man, and I was on one of his Zoom calls, and Dr. Warren Booth was on it too. I'll definitely reach out because it's definitely something that I'd like to know more of, because especially when it happens, and a lot of people ask me, it's like, hey, what's this mean? And I'm like, it just means that the female did that. Well, why? I don't know. I'd like to be able to answer questions, with a little bit of, scientific behind it, make me look smart with a bit of, few bigger words. That's what I need to do. Have some, I think when I say parthenogenesis, like, what's that? Like, most people don't know what it is. It's like, oh, you don't know what parthenogenesis is? Oh, yeah, I feel special. Oh, man. But it's funny when you hear you guys speak about stuff like just like language, blah, 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 and I can, and you guys understand it. And we're like, what? And just like, when I get so excited because I'm hyper, but when people say, oh, you breed ball pythons, tell me about it. And I'm like, oh, here we go. And then it's like, blah, 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 blah. And I'm talking, it's like, I don't understand anything you said, but man, you must love it because you're super excited. But, we speak a language that we all are commonly used to. And when other people that have no idea sort of come into that, they're like, what? So it's really neat how, when you guys talk amongst yourself, I'm just like, I think I know what you're talking about.
I have friends that fix cars, and whenever they try to talk to me about fixing cars, I'm like, what?
Yeah.
It just goes over my head. So
yeah, no, I hear you. I mean, it's great, but and it's great how then, at some time, somewhere, you link up and you unite and you cross paths, which is cool. So.
Mm-hmm. Yeah, I think that was what was really needed for this, like to bridge the gap between academia and commercial breeders. So yeah, it's awesome that it's finally being done. And I think we have a lot to learn, use all that wealth of genetic variation that's in the ball python trade.
Do you guys mind if we get some questions from the chat? That you guys have a little bit of time? And just before that, I do a little segment. What I do, I love to promote the hobby and people in it. I'll just take it away. I'll try to do it in 10, 15 minutes. Ready? Or less. Ready?
It's time for shout-outs of the week.
First clutch and two first egg cuttings. So the first time they've ever cut eggs. My man from Music City Balls, he had 5 eggs. And this is his first ever. So he's over the moon. So 5 eggs, vanilla, VPI exanthic, and he'll send a shit in to make sure that it is VPI, to a pastel. So he's making vanilla cream het exanthics. So congratulations, man. Welcome to actually producing eggs. It's such an amazing feeling. And that's so awesome. And then the first 2 egg cuttings. So people that have never cut eggs before, and that's something that I do if you don't know already. KC's Royal Pythons, black pastel, banana lesser times leopard pied. They got I'm not going to go into what they got, but they did okay. And then a pairing between Cali-Tex Exotics and Sybil Serpents, and they were incubated at Jacob's Place, Sybil Serpents. Super GHI, Super Pastel Lesser times Fire Pinstripe. Super cool. It's a nice pairing. And 500 on Instagram. Excuse me. Wow, I don't even have any water left, see? So 500 followers on Instagram. We have, wow, this is Teachers Pythons. That's what you know what, Heather, that's what yours should have been. Instead of ballroom, should have been Teachers Pythons. It was probably taken, but that suits you. Teachers Pythons, that's you to a T. But congratulations, Teachers Pythons, 500. We double that at 1,000. We have Boom Exotics, Swamp Water Exotics, and Bellamar Reptiles. Oh, and 2 others. The Royal Clutch and Rodney from Black Sun Reptiles. 1,500 on Instagram. We have Headstamp Ball Pythons. 2,000 on Instagram. We have KE Royal Reptiles, Kyle and Liz. Then 3K, we have Mazzotta. I'm going to go Mazzotta. I'm probably wrong. I butcher names all the time. Mazzotta Morphs, 3K. And then a big jump, huge jump. 100,000 followers on Instagram. The man, JKR, Justin Kabilka. Congratulations, man. Sick. And then last and final person, 2,000 on YouTube. We have DMJ Reptiles. Congratulations. And last but not least, Artist of the Week. It's Reptileremnants, Snake Shed Jewelry, Guitar Picks, and even Ornaments. Accepts custom orders, Instagram, website, and Etsy links in description. We just talked about that. See that Snake Shed Jewelry? It's so cool what we can do and what we're going to do. Imagine there's going to be stuff that we're going to be doing with snakes and stuff down the road. It's like, you know, it's going to be like, you know, if I said it right now, you'd be like, you're nuts, but it's going to happen like 10 years from now. It's crazy. So Billy needs a water break. Some of the people in the chat, people needs a water break. You know, Justin who? Come on, man. People. So anything before we get into some, you know, questions if people ask, anything else you guys want to, you know, add or say or talk about or, you know, I know, you know, Austin's been, you know, chatty, too chatty. The only time he woke up was when I said Ultramel. His favorite gene.
What are the top 3 morphs that you would like to have the genetic basis for, if you could right now? Not including the ones we know already.
100% Monsoon, because I've had Monsoon in my collection for 11 years, and I didn't know until the last 2. So I've been breeding animals. So I have probably 107, well, more now, but about a year ago, I had 107 animals that are minimum 50%. They came from the lineage. So I have 107 animals that are possible Het Monsoon. I know from some of the markers that I think that I know how to tell what Monsoon or Het Monsoon is. I think, you know, a good 60, 70% of them are. But that'd be one, because I could go now and instead of using a male, which I only have one visual male. Oh, and I might even have 2, but it's super special, probable leopard, possible spot nose, possible pin, possible pastel. You know, if that is a Monsoon, I'm going to have to prove it out. But I don't want to use him and go to something else and then, oh, I missed, or did I miss? So it would be nice to know what I've got, because I did a lot of het-to-hets, and I got like 4 eggs, 5 eggs, 6 eggs, and of course I miss. But with 1 in 4 odds, I can't say for sure that, you know, the 100% to the 50% isn't, you know? So I'd like to prove a lot of stuff out. But moving forward, I have a male that is visual that I'm going to breed, but should I use him to further the project and make some crazier stuff for the future, or do I use him to prove stuff out? Oh, like it's that huge debate, right? So that'd be one. Yellow belly asphalt. I know. I guess we're close on that. Pretty close. Can we divulge that? Did I screw up? Oops.
Well, we have DNA extracted from a bunch of ivories. But yeah, we just need more samples because we only have about 15 ivories DNA extracted from right now, and 4 of them are also have pastel, and we also have a super pastel pool, right? So we're gonna have to be careful of which individuals we sequence. Yeah.
So yeah, we'll work on that. But I would say asphalt yellow belly to tell the difference between that. Monsoon. What are we looking for, Austin? What's that other one that we could?
Clown?
He had clown. It's clown.
Yep.
Because there's no het markers for it.
Yeah, we're close to there, I think. So it's those are my 3. But I mean, I could sit here for the next 4 hours and name ones off because I want them all tomorrow. Check your. I'm already messaging you.
Yeah, from a scientist standpoint, well, from a scientist and a breeder standpoint, I'd like to know what's going on with the different lines of Exanthic.
Yeah, there's another good one, yes. But I only work with the 2, and I sort of know where they are. So to me, it's not like a priority. But that is, why aren't they? Why are they Exanthic and not compatible? Where albino and a candy and, there's some interaction, but they have none. I mean, I was there, and this was a true story. I had a VPI Exanthic male, and I got him a year after because I ended up getting 3 VPI Exanthic females. And I bred the VPI Exanthic to the 3 VPI Exanthics to make VPI Exanthics because they were very expensive back in the day. And I had 22 eggs from the 3 females, from visual to visual, and I got 22 normals. So I'm like, what? So I called the one breeder, and I'm like, dude, what's going on? It's like, nope, they're het. Where'd you get them? Like, I got them from this. And blah, blah. I'm like, well, I got my VPI Exanthic male right from VPI Exanthic. So I'm pretty sure that I didn't fuck up. And, I'm like, dude, when you do visual to visual with 22 eggs and you don't hit a visual, it's impossible. They're not VPI Exanthics. So I had 22 double hets. And I think I was like 17-5. Like, it was just like, could it get any worse? But yeah, double hets. Crazy. But that would be an amazing thing to find out, to see why they aren't, and to, because I wonder if some people think that they have Exanthic, VPI Exanthic, but it's SK or, I think they're pretty diverse, though, that you can tell without having to test them what they are.
And I say one of the reasons why I say from a scientist perspective, so like we know a lot about the mechanism that causes albinism, even the different types of albinism, but because reptiles have a different color mechanism than us, like they've got 2 different simplifying it, 2 different types of pigment cells that determine whether or not, and that's why the albinos don't come out all white, because they still have cells that are producing pigment. And so it'd be cool to kind of figure out, well, what's the mechanism that's causing the yellow to not be produced in those particular animals.
Yeah.
So,
yeah, like a gene that regulates all non-melanin pigmentation, right? The pteridine, carotenoids, that sort of thing. Yeah. Yeah, I agree.
See, it's amazing how this is interesting for, a breeder, just for our industry, and then scientifically and even educationally, this is something that everybody's excited in all these different fields for different reasons with the same animals. I think it's super cool. We're united because of these snakes, and it's awesome. I would never have met any of you if it wasn't for snakes. I mean, how crazy is that? I mean, it's super cool.
We converged on the interest, right? Genetics of ball pythons, and yeah.
You don't have a snake now, do you, Alan?
No, I don't, but I did have one before I moved to Montreal. When I moved to Montreal to do my grad studies, I had to give them away.
We're gonna hook them up. We're gonna hook them up, and we're gonna make ball python at least owners out of you. Maybe one day breeders, but at least owners.
Sounds good. I'm in.
That's awesome. Oh, here we go. Nice. Show and tell. Look at that. Like, that's so sick. Oh, is that a...
Okay, here we go.
Yeah, see, there you go.
He's a piebald. She is not
He
a
must have
piebald.
gone in.
No, he's not.
Did a sample of her? Oh, she's not a piebald. Oh.
No. I thought she's an ivory because, and she's a paradox or something. So anyway, someday we will know the answer. But yeah, and
Very
she
cool.
got sort of, you know how some of those have the split. Sorry, her name is Chaos. Yes, and she was, yeah, she's.
You know what that is? That is a paradox het pied. Or it's
there's no pied in.
not even het pied?
No.
Because that pattern looks het pied, doesn't it, Austin? Or
No, there's no. I've done this clutch 3 times. So it's just I don't
so
know.
sick. I love that. Like, that's so cool.
I don't know, Hannah. You can do your hypotheses, but the others in the clutch were ivories, but she's very clean for ivory. So there's something going on where, yeah, you can hypothesize.
So when you send her to me?
Yeah.
That's so super cool. I love it. Oh, you know what's also neat? I have one that's not so dramatic like that, but I have one that people swear is a pied, and it's just a paradox het pied. But the ringer on it is so massive. They're like, dude, that's a pied. I'm like, it's not. I know the pairing. It came out, and it's just sick. But you know what else is cool? Austin noticed it back in 2018. I actually hit. We call her. It's a him. What's that?
It's a chimera.
That's a chimera, yeah, but we call it Two-Face. So I actually produced a snake, and Austin's like, dude, look, you gotta, and I'm like, no, I don't. He goes, look at this thing, and every time it shed. So I have a snake that is het pied and het for, and this is what's interesting, het pied and het enhancer. So on one side, it's almost like a visual enhancer. You can almost see the enhancer in it. And it's directly straight, like half the snake is one gene, and then the other snake, the other half is het pied. But what's really cool about it is you look at one side, you see like the normal, it looks like a het pied with the markers. Oh, and that one side of the het pied has the marker and you know what we look for het pieds, but on the other side, it doesn't have any markers. I think that's super cool. What would cause an animal to show 2 different traits, 50-50? Like, is that even something that you don't know yet? Or theory?
I would hypothesize it's 2 snakes that fuse together. I don't know.
Yeah.
I mean, that's
What do you mean fuse together?
That's called a chimera.
Right.
They fuse together.
But how does it fuse together? Because tell me how to do it so I can make more. Because I want to make a pied clown chimera, like so sick. Not only that, I mean, it'd be the coolest animal to have, but people would be paying like $50 to $100K for those things. That'd be the sickest thing in the world. Show me how to do it.
What doesn't make sense about the snake is that half of it visually shows a recessive morph.
Right.
There's 2 different recessives paired together.
Yeah, which is nuts. So yeah, that's another thing. Good call. So it was from a visual pied to a visual, so an entry pied actually to an enhancer. So the only combination that could come out is entry double hets or double hets. I hit an entry double het, which just looks like a normal entry with the het pied marker and then the other one, you actually can see the enhancer. So it's an actual enhancer on one side and then het pied on the other.
I think you probably had some type of somatic recombination.
Can I reproduce it? How do I do it? Come on, show me.
Probably not.
Come on, tell me, how do I do it? Super cool though. I mean, if you could reproduce that, it'd be nuts. It'd be just crazy. Imagine half pied, like a half pied or something really cool like half ivory and like half freeway or something like, it'd be just super nuts. Or even reproduce that amazing animal that she won't let go. She's holding it because she knows I want it so badly. She's probably going to show it off and say, look, Billy.
You should get the chimera.
Actually, I am here. But no one wants it. I'll show it on Instagram. We got probably some questions here.
Your example reminds me of these chimeras that people have made between hamsters and gerbils. where you basically can mix the early embryos together really early, and you can end up with an animal where literally chunks of the body are hamster and other chunks of the body are gerbil, but they're similar enough body plans that it's compatible.
We got another clown in the. You know what, Callie? I'm gonna make you a moderator so that you can like.
Well, Billy, I really enjoyed this podcast, but I need to sign off in just a few minutes. So I
For
just
sure.
want to say how much I enjoyed being here and talking to everyone. And Alan and Heather, it was really good to get to meet you guys a little bit more as well. And
Awesome.
Yes, anybody listening, please send us sheds. I will email you the links so that you can post those along with this podcast.
Awesome. Yeah. So we'll wrap it up then. And then that way, it's a nice early night. I know that the Habs are you keeping? Are the Habs winning?
Yeah, it's 1-0 last time I checked.
1-0 Habs.
Yep.
Awesome, man.
End of the first.
Come on, Carrie. Keep the donut, man. Keep it 0, baby. Don't let any in. Come back with game 4 not hat trick. Jesus. Oh my God.
Keep the dream alive.
Yeah, but no. Thank you, everybody. Thank you for your time. It was very interesting, and I'm sure we'll talk back and forth. And if you can email me, Hannah, all your info, and then I can put it down below in the description. And people will message me and talk, and I have a lot of people in the U.S. too that I can basically say, hey, you know what, you should do this and help out and stuff like that, and even further down the road. And I guess Alan and Heather, we can talk more, and you should have a package of a bunch of cool snake sheds. Do you get really excited when you get them? Like, we just have them. We just normally throw them up. We package them. When you get them, do you get excited, Hannah? And it's oh my God, yay! It's
I especially like the little letters that people will send in. They tell me all the details about their animal. They'll say, here's some photos just for fun. So I really enjoyed that.
Super
part
cool.
of interacting with everyone who sends us sheds. That's very, very fulfilling.
That's amazing.
And if down the road, once we make more discoveries, say once we discover the clown and we have like a preprint ready, if you want to invite us over to kind of talk about the results and maybe explain them to your audience, I think I'd be happy to.
Cool. Yeah, definitely. I mean, it's definitely something that's awesome. And I'll work on sending more sheds and getting some other people in and working on that for our research up here. And, stay in touch, be safe. It was a pleasure having you all on. And, all of us keepers, we got to, convert these two. You do what you can in Michigan, and we'll do what we can here in Canada to convert these two, at least be owners. And it was a pleasure having you guys on. And I'm sure, it was very interesting. A lot of people are saying, man, this is awesome. And I know that this is just the first of many. And, every 6 months, we're going to actually have something better and newer and crazier. And I just think it's awesome what you guys are doing. And keep it up. And I appreciate your time, guys. And we'll say bye to everybody in the crowd, the chat. Thanks for showing up, and thanks for. They're gonna cry because this is gonna be the shortest one ever. Because as you can see, I talk a lot, and I just ramble, and I go into all different things. So they're like, "Oh my God, what am I supposed to do? I'm usually here for another hour or two." Sorry, guys, can't always get what you want. But thank you, guys. You guys are awesome. Austin's probably going, yes, I finally get out of here before 10 o'clock. But thank you, Hannah. Thank you, Chiron. Thank you, Alan. Thank you, Heather. And even you all, thank you too, Austin. Didn't do much, But anyways, thank you, everybody. Be safe.
Thank you for having us on.
Yeah, my pleasure, honestly. And it was a pleasure meeting you both. And everyone have a great night. And we're out.
Go Habs.
Happy Monday. Good night, everybody.
My channel. My channel. My content. My channel. My content. My channel. My content. Good night, all.